Hello friends, I'm Dr. Anil Nashinge. Welcome to this video. Friends, in this video you are going to learn about how to prepare a culture medium. So the culture media, they are the essential nutrients which we supply for the growth of the microorganisms, and the culture medium, it contains all the essential nutrients and the raw materials. Because in the microbiology laboratory we grow the microorganisms, we cultivate the microorganisms, and for the growth of microorganisms, for the cultivation of the microorganisms into the laboratory, we have to provide them with an essential nutrients. So the bacteria, they vary in their nutritional requirements. So the nutritional requirement of all the microorganisms, it is not the same. So they will vary with the type of nutritions which are required. Microorganisms, they need carbon sources, they need nitrogen sources, they need various elemental requirements, and we have to supplement, we have to provide all these essential ingredients so that the microorganisms will grow by utilizing all these ingredients, and then we can grow these microorganisms in the laboratory. So there are different types of the culture media. Depending on the state, the culture media, they are classified into two types, that is solid medium, liquid medium, and there is also semi-solid medium. The broth, it is called as the liquid medium, and the solid medium, it is containing solidifying agent, that is agar, and another one is the semi-solid medium. The nutritional requirement of different bacteria, it is different, and a single culture medium, it will not be sufficient to grow all the kind of microorganism, because the different microorganism, they vary with respect to their nutritional requirement, and we have to design the culture medium in such a way that so that culture medium, it will provide all the essential ingredients, so that will allow the growth of all kind of microorganisms, and the culture medium should be provided for the growth of microorganism. And in this video we are going to learn about how to prepare a culture medium. So first of all, we are going to discuss about the preparation of nutrient broth. How will you prepare the nutrient broth? So we'll see how to prepare the nutrient broth. So friends, in the market, a ready-made available nutrient broth is supplied by the suppliers. We can get it from the market, and this nutrient broth, it is a type of liquid medium, and here we can see that it is a readily procured, ready-made available nutrient broth, and that nutrient broth, it is used for the cultivation of microorganism. So the nutrient agar and nutrient broth, they are a general purpose nutrient media, and they are used for the cultivation of a wide range of microorganisms. So different types of bacteria and a wide range of non-fastidious microorganisms, they can be cultivated into this nutrient broth and nutrient agar, because this nutrient broth and nutrient agar, it contains all the ingredients. So whatever the essential ingredients, they are present, they are supplied by this culture medium. So it contains peptone, it contains beef extract, it contains NaCl, it contains agar, and we have to add all these ingredients into appropriate amount of the distilled water. So today we are going to see how to prepare this culture medium, and it is an excellent culture medium. It is found to be one of the ideal and the excellent culture medium for the growth of a wide variety of microorganisms, because all the essential ingredients, they will be supplied by that culture media, and we can grow these microorganisms. So we are going to see how to prepare a nutrient broth and how to prepare the nutrient agar. This nutrient broth and this nutrient agar, they also use for the isolation of microorganism, for the cultivation of microorganism, for the maintenance of the microorganism, and they can be used for the enumeration of microorganisms which are present in air, which are present in the food, present in the dairy product, in the sewage, and in the different sample we can enumerate the microorganism. So let's see how the nutrient agar and nutrient broth, it is prepared. So first of all, we'll see the preparation of nutrient broth. The nutrient broth and nutrient agar, they are of the same composition. Only the nutrient broth, it is the medium which is devoid of agar. So the agar, it is used as a solidifying agent. It is not added into this nutrient broth. So it lacks the solidifying agent, that is agar, and in the nutrient agar there is addition of agar. So agar it is used as the solidifying agent. So let's see the procedure of nutrient broth. Friends, now we'll see the procedure of how to make the nutrient broth. So as I told, the nutrient broth, it is the readily procured from the market. We can get the ready-made powdered form of the nutrient broth, and we can directly prepare that nutrient broth. So onto this nutrient broth, that is the type of liquid medium. So we can see there is a label that is of the company High Media, which is the standard, and there is the label and that have shown the directions for the preparations of the nutrient media. So here we will see the composition of nutrient media. So nutrient broth, it is consists of the peptones, sodium chloride, beef extract or yeast extract, and then we have to add 1000 ml of distilled water. pH should be adjusted to 7.4, and the nutrient broth, it is containing all the essential nutrients. It contains the source of carbon, the source of nitrogen, and all the essential nutrients, they are present into this nutrient broth. So we'll see the procedure of the preparation of nutrient broth. So here it is given that peptone 5 gram, sodium chloride 5 gram, meat extract or beef extract 1.5 gram, yeast extract 1.5 gram, and the pH we have to adjust 7.4, 1000 ml of the distilled water, and this is the readily available nutrient broth. Okay, readily available, and here the directions to prepare, they are given. So you have to suspend 13 grams in 1000 ml of distilled water, and you have to heat it to dissolve all the contents completely, and then you have to dispense it into tubes or flask as per our requirement. Then we have to sterilize it by autoclaving, and after sterilization at 120 degrees Celsius, that is for at 15 lbs pressure, we have to keep it for sterilization for 15 minutes, and then we have to cool it, and then we can use it. So that is the readily available nutrient broth bottle. So we can prepare this media as per our requirement. So we are going to prepare 100 ml of the nutrient broth, and for the 100 ml we need 1.3 grams of this readily prepared available nutrient broth. So we can take 1.3 gram of this powder into, and we add it into 100 ml of distilled water, and for 1000 ml we have to take 13 grams. So we'll take 1.3 gram of this powder. Now we are taking 1.3 gram of the nutrient broth powder. So we have to weigh it accurately. So by the digital balance, we have to weigh it accurately, and we will take 1.3 grams of this powder. We'll add more. Now we have weighed accurately 1.3 gram of this powder of the nutrient broth, and we will add it into 100 ml of the distilled water. So we'll take 100 ml of the distilled water. It is very essential for the preparation of the culture medium. We should not use tap water. Only distilled water should be used for the preparation of the culture medium. And then we will suspend that 1.3 gram of the accurately weighed nutrient broth powder into the 100 ml of the conical flask, and then we will mix it properly so that all the ingredients, it will be dissolved. Then after mixing, we will put it onto the hot plate. So we will place it onto the hot plate for the boiling or for the heating all the contents. So we have to boil it, we have to heat it for a few minutes so that all the ingredients they will be dissolved. So we have placed the nutrient broth onto the hot plate to dissolve all the ingredients completely. So we have to heat it while stirring it properly so that all the ingredients will be dissolved. Now we'll take it. Okay. So we have plug with the cotton. So we can see that the conical flask, it is labeled with the nutrient broth. We have to label it. We can mention the name of that culture medium and the date. We can also mention the class, so that labeling it is also very essential. And then we have to wrap this. So we have to wrap the conical flask with the paper, with the aluminium foil, and after wrapping, then we have to place it into autoclave for the sterilization. So autoclave, it is the instrument which is used for the sterilization, and we have to keep this culture medium into the autoclave at the temperature of 121 degrees Celsius, 15 lbs pressure, and time period 15 minutes. And there will be the proper sterilization of the culture medium. So we can sterilize all the culture media into the autoclave, and after proper sterilization, after a proper holding period of time, then we have to remove that culture media, and then we have to cool it, and then we have to dispense that culture media into the test tube. We can dispense that culture media into the different flask according to our requirement.